सर। ये बता दें। Photographer आए थे। Hello. Hello. Hello.
Saraswati Vandana is right now.
Right now. I give them time to settle down.
Good morning, everyone. We're delighted to have you all here. May I request all of you to please keep your cellular devices on mute as we'll be starting momentarily. Please settle down.
नीचे वाला टेबल तो रखना है दो फिक्स से
Don't make it up. Yes, pose for it. Kindly settle down. Morning, everyone. I'm delighted to have you all here. I, Iba,
And I, Sneha Bajpayee.
are absolutely delighted to be your host for this special day. It gives us immense pleasure to welcome you to the 16th Foundation Day of THSTI. A very warm welcome to all the dignitaries, guests, faculty, and colleagues present here today. It's a truly proud moment as we come together to commemorate 16 years of THSTI's journey, driven by innovation, collaboration, and a relentless pursuit of excellence in translational research. From cutting-edge translational research to path-breaking public health solutions, THSTI has emerged as a beacon of scientific excellence. And today is all about celebrating that legacy.
To start this auspicious day for THSTI, I now request Senior Professor Dr. Nitya Wadhwa to give the welcome address. Good morning. It is 16th Foundation Day of the Translation Health Science and Technology Institute. We are honored to welcome our Foundation Day speaker, Professor Anil Kaul. We appreciate your insights and time, sir. A very welcome to our esteemed guests from the academia and industry, our valued collaborators, my colleagues, and of course our students and research staff, the heart and soul of the institute. As we gather to mark 16 years of THSTI's journey, it is a moment of reflection, pride, and renewed purpose. We remember with respect and affection Professor M.K. Dhan, whose visionary leadership gave birth to THSTI. His building bridges between science and society remains at the core of our mission. His legacy lives on in every program we provide and every partnership we forge. Over the last 16 years, THSTI has evolved institute. From foundational discovery to clinical and public health applications, we have strengthened the health ecosystem through innovation, partnership, and scientific excellence. The past year has been especially transformative. We have created infrastructure, the Medical Research Center, and the upcoming translation research facility designed to enable end-to-end translation research. These facilities reflect our aspiration to scale impact and support India's journey toward health innovation. Our Foundation Day is also a time to look ahead. Just yesterday, Singh 2025, our second industry academia meet. Industry participants dialogues, the event became a springboard for translational partnerships aligned with the Government of India's Bio-P3 initiative. A sincere thanks to all who made it a success. As we move forward, our commitment remains steadfast. Cutting edge science, foster meaningful collaborations, and develop health solutions that matter for India and for the world. We express our deepest gratitude to the Department of Biotechnology and institutes like ours thrive and run well in the system. Once again, for celebration of our journey so far, but also sparks new ideas, new relationships, and a shared sense of purpose. Thank you. Ladies and gentlemen, as we continue celebrating 16 years of translational excellence at THSTI, it's time to dive in to what truly fuels our collective journey, science. We now move into the heart of today's event, the Scientific Colloquium, a platform where ideas are exchanged, frontiers are challenged, the spirit of collaboration is celebrated. This colloquium brings together some of the finest minds from our own community and beyond, scientists, clinicians, and innovators. Exemplifies the kind of impact-driven work THSTI stands for. Today, it's the symphony of scientific insights from bench to bedside, from molecules to mechanisms, and from questions to possibilities. So without a further ado, let's begin with our first speaker, Ms. Bhavna Verma, is going to gut microbiome dysbiosis, T-cell response during experimental colitis.
Good morning, everyone. I am Bhavna, a fourth-year PhD student working under the supervision of Dr. Amit Awasthi. Today, I will be presenting my doctoral work titled Effect of Gut Microbiome Dysbiosis on Host T cell Response During Experimental Colitis. First of all, I would like to briefly talk about inflammatory bowel disease. Inflammatory bowel disease is an inflammatory condition which is caused by aberrant immune cell reaction against the gut microbes present inside a host. This chronic inflammation leads to the disease condition. Depending on the part of gastrointestinal tract that gets affected, it has two major types. First one is the ulcerative colitis, and in ulcerative colitis, the submucosa and mucosal rectum is affected, and there is some varying degree of lesions observed in the colon. And in Crohn's disease is a segmental and a transient form of disease which can affect any part of the gastrointestinal tract, but it primarily affects the perineal ileum and the beginning of the colon. So, why do we need to talk about this condition? This figure shows the global incidence and prevalence rate of IBD among the people of different age group. But we also need to look at the Indian perspective of IBD. So this map A here shows the state-wise incidence rate of IBD in the year 2019, whereas this map B shows the state-wise incidence rate which has changed from the year 1990 to the year 2019. So it is conclusive to say that long gone are the days when IBD was considered the ailment of the Western world. And with the current therapeutic options available, the patients are barely able to manage the disease, which is accompanied by lifelong morbidity and strongly reduced quality of life. So we need to identify different therapeutic options. Now, what causes IBD? The primary etiological factors of this disease involves genetic predisposition of patients, environmental factors, and gut microbiota. In this study, we are trying to correlate the gut microbial dysbiosis with host immune cell response. Now, before diving deep into this, I would like to briefly mention about the human microbiome. Our body harbors huge array of microorganisms. While bacteria are the key players, other single-celled organisms like fungi, archaea, and viruses are also present. Different organs like the skin, oral cavity, vagina, and the gut have their own distinct microbiome. Out of all of this, gut has the highly diverse microbiome, which is mostly studied. As I mentioned, in the patients suffering from IBD, there is often the gut microbial dysbiosis is observed. So, what is dysbiosis? Dysbiosis is defined as the imbalance of the gut microbiota, which is accompanied by either loss of symbionts or outgrowth of pathobionts. As I mentioned, the bacterial are the key players of gut microbiota, and the researchers have widely explored this bacterial dysbiosis in various disease settings. But as I mentioned, other single cell organism, although present in low proportion, has a significant role to play. Particularly the gut fungi, and here I am mentioning it as the gut microbiota. So upcoming researchers have shown that gut microbiota is also important in various disease conditions and have explored them in various disease settings. And they are not the mere bystanders, although present in low abundance. So to start with, we started to explore the gut fungi dysbiosis using a model of experimental colitis known as DSS-induced colitis model. In this model, we created mice which were gut fungi depleted by subjecting them to broad spectrum antifungal drug in their drinking water. Once the depletion was confirmed by checking the expression of internal transcribed spacer region, we found that, and we started that fungi has depleted completely, and then we subjected these mice to DSS. Now DSS is a region which is toxic to the surface epithelium, and upon drinking this DSS dissolved water, mice develop colitis.
So we found that the mice in which we depleted the fungi beforehand, they suffered from a severe disease. Upon harvesting the colon from these animals at the end point of the experiment, we found that there is significant shortening of colon length in the fungi depleted group of the animals. Also, when observing the probability of survival, we found that the probability of this survival is less in AF plus DSS group. We also did the pathological evaluation using HNE staining and found that there is indeed more ulceration and immune cell infiltration in the AF plus DSS group compared to the DSS alone group. We also wanted to check the immune profiling. We did the immune profiling of the cells isolated from lamina propria cells of the intestine and found that there is significant increase in CD4 positive, IL-17 positive T cells and an upward trend observed in IFN gamma positive cells. But this DSS model has a limitation. We can only check the TH1 and TH17 effector functions and what role they have to play in the gut microbial dysbiosis. So we explored another model of experimental colitis, which is called oxazolone-induced colitis model. In this model, what you have to do, you have to pre-sensitize the animals with a higher dose of oxazolone epicutaneously. And on the day 5, you have to again challenge the animal with a lower dose of oxazolone and subsequently the colitis is induced. So in the similar experimental settings, we did the experiment and found that indeed in the gut fungi dysbiotic mice, there is more disease and more weight loss, and even the survival is less in these animals. In this model, there is no significant shortening of colon length, but you have to look at the lesions which are forming in the colon after the disease has occurred. So there is more lesion formation, but in order to confirm that indeed these mice have more severe disease, we did the immunopathological evaluation. And yes, it was found that there is more inflammation and immune cell infiltration in AF plus OXA group. Also, we did the immune profiling of mesenteric lymph nodes and found that, yes, there is more CD4 positive, IL-9 positive cells and significantly increased IL-4 positive cells as well. So while we were performing these experiments, a paper came out in Nature Medicine, which has taken the PBMCs from ulcerative colitis patients and CD patients and these cells were found to be reactive towards the lysates they have isolated from different fungi and bacteria. But the highest reactivity was observed towards the lysates which were obtained from the Candida tropicalis and Candida glabrata and Saccharomyces cerevisiae. Not much difference was observed for the Candida albicans and also for the more phylogenetically distant fungi like Melissasia restricta and other bacterial lysates. So keeping this in mind, we designed another experiments where we started to colonize animals with specific fungi. And the fungi, the two fungi species we take were Candida parapsilosis, because this species was not explored in the study I just talked about. So we took this species and colonized in the animal, and as a control, we take Saccharomyces cerevisiae because it is readily available in bakery products and is known to cause inflammation in the animals. So after successful colonization, we subjected these mice to oxazolone-induced colitis, and interestingly, we found that the mice which were colonized with Candida parapsilosis were kind of protected from the disease, whereas the animals which were colonized with Saccharomyces cerevisiae, as expected, had more severe disease. Again, the lesion formation was observed, and there is less to none lesions were observed in Candida parapsilosis colonized group of animals compared to the Saccharomyces cerevisiae colonized and the control group. Now we did the pathological evaluation, and again we found that there is more inflammation and infiltration of immune cells in the Saccharomyces cerevisiae colonized group of animals, whereas the mice seems to be protected from this inflammation and infiltration in the Candida parapsilosis colonized group. When we did the immune profiling, we found that there is less percentage of cells, significantly reduced percentage of cells of CD4 positive, IL-9 positive cells and CD4 positive, IL-4 positive cells. Since this was an in vivo model, we wanted to check that whether this Candida parapsilosis is specifically acting on these TH9 cells. So we performed an in vitro experiment using a TH9 differentiation assay, which is well standardized in our lab. And one group of cells were co-cultured with the Candida parapsilosis lysate, and one group of cells were co-cultured with Saccharomyces cerevisiae group of lysates. And it was found that the CP lysate was able to reduce or regulate the generation of IL-9 cytokine from the TH9 cells, whereas no effect can be observed in the cells which were cultured with Saccharomyces cerevisiae lysate. So we also confirmed this IL-9 expression using RT-PCR and found that yes, the data was correlating with what we have observed with the flow cytometry data. To summarize, I would like to say that when we create gut fungi dysbiosis there and subject these mice to colitis induction, because of increase in the TH9 population, there is high inflammation and the severe colitis occurs. But when this can be rescued by colonizing the animals with Candida parapsilosis, because this fungi is directly acting on TH9 cells and reducing its number, causing less inflammation and leads to colitis amelioration. Future perspective of the study is, this study shows the correlation between the TH9 and gut microbiome during colitis, which is first of its kind. But both Candida parapsilosis and Saccharomyces cerevisiae are yeast, but their differential role in disease progression and TH9 cell modulation needs to be further assessed. Targeting the TH9 microbiome axis may complement existing IBD therapies by developing engineered probiotics to suppress TH9 mediated inflammation. Thank you. That's all. I would like to thank my supervisor, Dr. Amit Avasthi, for his constant guidance and support. The funding agency, THSTI and DBT for my fellowship, and the facilities provided by THSTI, experimental animal facility, FACS facility, histopathology facility. I deeply acknowledge my lab members and my fellow scholars, and a big thanks to every member of Immunobiology Lab. Thank you.
Thank you, Ms. Bhavna. Next up, we have Mr. Rohit Kachwaha, who will be delivering a colloquium on microRNAs as key regulators on placental development and pregnancy complications.
Good morning, everybody. I'm Rohit Kachwaha, a fourth-year PhD scholar at THSTI in Dr. Pallavi Kshetrapal's team at Lab of Perinatal Research. So the presentation is about a small part of my PhD, which is about microRNA, exploring the roles of microRNA as key regulators in the placental development and the associated pregnancy complications. So let's talk about the keystone organ of pregnancy, which is placenta. We have been hearing the functions of placenta like old world tales, and we are not oblivious of the fact that how much placenta is important for the development of baby, as well as when the gestation period is going on. Throughout the decades, it has been found out that dysregulation in the structure, or so much to say the structural deficits in the growing placenta can lead to functional dysregulation that in turn leads to pregnancy or obstetric complications. If we talk about dysregulated molecules in placenta, many molecules are known. There is a repertoire of molecules, out of which microRNAs are those 19 to 20 nucleotide long, notorious, or you can say beneficial oligos which are there present in the cells. We found out that myriad of microRNAs are associated with various pregnancy outcomes, which are mentioned here. Many of these studies are descriptive, so that intrigued us that how the mechanistic role is being played by these small oligonucleotides inside the cell. If we talk about preeclampsia, the disease which we wanted to study and where our candidate microRNA was picked from, which we'll be talking about in the further slides, let's just look about preeclampsia, which is a hypertensive disorder. Preeclampsia has been known for centuries. So, and more or less, the clinical manifestation of preeclampsia has remained the same. It's a hypertensive disorder which is characterized by systolic and diastolic blood pressure of more than 140 and 90 millimeters of mercury, and release of excessive amount of protein in the mother's urine, which is more than 0.3 gram per 24 hours. As a long-term outcome, the mother is affected due to organ damage, and there are many more clinical symptoms, headache, blurred vision. If we talk about the epidemiology of placenta, there is about 2 to 8% of worldwide prevalence. However, in the North Indian Garbhini cohort, we have observed the prevalence of around 4.3. However, what's the burning question that how it leads to maternal morbidity and perinatal morbidity, as well as with long-term ill effects of the complication. So we deduced a hypothesis where the placental microRNAs will be regulating such functions that are important for the placental development, which, if dysregulated, can lead to placental-mediated hard pregnancy outcomes. We deduced an objective where we wanted to evaluate the function and the effect of these microRNAs on the growing placenta. We picked a few data sets which are publicly available on GEO, and we combined them together to find out which significant microRNAs are known on the public level. We found out there was a repertoire of microRNAs again, but we had to short them down. We found out across the combination of at most 3 and at least 2, there were 29 microRNAs, again, which was very hard to work with. So we reduced these microRNAs from 29 to 10 using an approach where the microRNAs only expressed from chromosome microRNA cluster 14 and 19 were picked. These are 2 well-known microRNA clusters. So the number was reduced from 29 to 10. Now, how do we check which microRNA could be more interesting to work on and which one has not been studied thoroughly? First, we tested the expression of all these 10 microRNAs in placental cell lines. Before we establish an in vitro model, we took JEC3 cell lines and H3R8 cell line and also tested the expression of this microRNA on using a term placenta sample. We found out that the basal expression in comparison to the U6 control. We picked microRNA 5223P, HSA microRNA 5223P. In the dark boxes that we see on the upper part of the slide are actually the roles of microRNA that are known in the cancer. It's well studied in cancer. However, if we look at the yellow boxes down at the slide, in placental studies, there are various platform studies where, as a result of sequencing, this microRNA has been elucidated. And in one of the studies, which has been published in 2022, it was an Italian study where this microRNA was found to be upregulated some 1.73 folds in the preeclamptic placenta. Now the question was the mechanistic study of this microRNA in the placenta was not known. So we thought, okay, fine, then we can go with this microRNA since placental studies are not known. We first established a model using a placental JEC3 cell line where we mimicked the expression of microRNA using 40 micromolars of mimic construct, as well as we downregulated the expression of microRNA using an siRNA construct at the same concentration. We did observe that upon mimicking, we can see in the yellow bar, the expression has went up, and upon transfecting an siRNA, the expression of microRNA has went down. This was validated with this MTT assay, which ascertained us that there is nil cytotoxicity on the cells. Now this model was established in vitro. We thought, let's now proceed and look at the functions of this microRNA in the context of placenta. If we look at this diagram, from top to bottom, the placenta develops the cytotrophoblast that we see in the yellow, these yellow cells. These cells differentiate and they come down into the decidua of mother's uterus, where these cells migrate. And there are two major events which we will be looking at in this presentation. One is differentiation, where the cytotrophoblasts differentiate into syncytiotrophoblasts and subsequent cell types. So the key pathway that we observe here is first differentiation, which is marked by genes like GCM1, PLGF, and so on. Now, expression of this microRNA that we have observed in the angiogenic pathways as well. Since we understand neoangiogenesis is utterly important for the development of the placenta, from where the developing baby draws blood from the mother. All right, moving forward, interestingly, we found out that genes like Notch1, Wnt, and PLGF were downregulated, which are especially known markers of placental development. However, GCM1 was found to be a predicted target of this microRNA scan, which was also downregulated. Now, again, this was a predicted target, so we decided to go ahead and look at the protein expression of this gene, and the trends were similar. We moved ahead whether to ascertain this gene GCM1 is a direct target of this microRNA. We performed a functional analysis, so much so to ascertain the mechanistic involvement of this microRNA with this gene. We did observe upon performing luciferase reporter assay. So the agenda was, if there is a perfect binding of this microRNA to the 3' UTR region of the gene GCM1, we expect that the decrease in the bioluminescence, which was shown by the decreased RLU units when we compared to the control. This pointed that GCM1 could be the direct target of this microRNA. Moving ahead, when we were giving you a short recap when I was talking about that how cells will differentiate and then they have to migrate into the residue of placenta, we checked on the cellular level whether this microRNA has a role on the migration of the CHEK3 cells. We performed transfer assay as the validatory assay, where the cells were allowed to migrate in avoidance chamber from depleted media to a complete media. We did observe, upon mimicking this microRNA, we can see in this, we did observe when compared to the control, the migratory potential of the cells, CHEK3 cells, have been reduced. We can see in both 4x and 10x, which is marked by the decrease in the absorbance upon mimicking of this microRNA. So this points out that this microRNA impairs the migratory potential of the CHEK3 cells. Moving further, if we have to talk about a normal pregnancy, in normal pregnancy, it's known the PLGF pathway, which is placental growth factor, is upregulated, which it should be. In denomination, when compared with SFLT, which is the solute FMS-like tyrosine receptor, the ratio goes down, which essentially is actually an indicator of a normal pregnancy. However, what we observe that there is a disrupted ratio where the SFLT and PLGF ratio has went down. Recall from the differentiation slide that the PLGF levels upon the mimicking this microRNA has went down. Again, that we see the ratios has went up, which points to the elevated levels as we see on the clinical levels in preeclampsia. We also found out a novel association of this microRNA 5223B with soluble endocrine by TGF-beta, where again there was a pathogenic increase in the levels of these molecular markers. However, it is still elusive that we are actually working on whether or not this microRNA can be used for an early risk prediction or can be used for therapeutic intervention. Just to summarize, we see that upon mimicking of this microRNA that we have phenocopied in the placental cell lines, which was found out to be elevated in the preeclamptic placenta, we observed that there was a decrease in differentiation, which is observed by genes like GCM1, TGF-beta, Notch1. Because of the shortage of time, I'm just presenting the summary of the slides. But we also have tested the apoptotic genes where caspase 3 and caspase 9 were predicted targets of this microRNA, and they went down, somewhere affecting the apoptotic pathways in the CHEK3 cell lines. Also, when we talk about migration, migration was impaired due to the increase in the expression of this microRNA, as well we see due to which angiogenetic pathways were also affected. So we can ascertain, at least for now, that there is an etiological link between this dysregulated preeclampsia associated markers and this microRNA. With this, I would like to conclude and thank my principal investigator, Dr. Pallavi Kshetrapal, and the Lab of Perinatal Research, along with Garbhini team and all the lab members and friends. Thank you so much.
Thank you, Rohit. Now we have Ms. Sandalia presenting her presentation titled Harnessing Glycovariant Signatures to Improve Specificity of Tumor Markers.
Hello everyone, good morning. I'm Sandalia. Today on the occasion of 16th Foundation Day, I'm presenting my work which is about harnessing glycovariant signatures to improve specificity of tumor markers. We know that cancer is the abnormal proliferation and acquirement of metastatic properties by a normal cell. Given that cancer has been the cause of more than 10 million deaths in 2020 alone, it's one of the major causes of deaths worldwide, and it's mostly attributed to lack of early diagnostic markers. Currently, diagnosis is majorly mediated by a combination of imaging, biopsies, or serum tumor antigens. But as we can see, these methods are not reliable, especially for early diagnostics. Let's focus on tumor marker detection tests. These tests traditionally target glycoproteins and are one of the first tests performed in the process of diagnosis. But a major issue is that these markers are also noted in benign conditions. It largely affects the sensitivity and specificity of the assays. If you add this to the lack of symptoms during early stages of cancer, which is common for many cancers, early diagnosis is simply not possible with any of these. And for individuals who are at high risk, who need to get screening regularly, it becomes even more challenging to catch cancer early on because waiting is not an option. But why are these markers like this? What is the reason behind this lack of specificity? If we take a closer look at how tumor markers are detected in the assays, traditional methods often do total protein detection. That is, only the protein part of the glycoprotein marker is targeted by both the capture and tracer antibody, as we can see on the left. There are a couple of assays which do total glycan detection as well, but they follow the same format. The target of these assays is not cancer specific. The marker is elevated in cancer, sure, but the format cannot distinguish between marker from a benign state and from a malignant state. So how do we change it? One way is to improve the specificity by targeting the glyco part of the glycoprotein tumor marker. That is by targeting altered glycosylation. But first, glycosylation. So see these basic sugars that we are familiar with, glucosamine, mannose, fucose, and the likes. So these monosaccharides are present as complex carbohydrate chains within the cell, and they are known as glycans. And glycan-containing proteins are called glycoproteins, which is like the tumor markers I just talked about. So when we say glycosylation, it's a very common occurrence in cell, the attachment of these glycans to proteins and lipids. And it's a very normal phenomena. But certain conditions can disrupt the native glycosylation patterns in cell, like cancer, and generate altered glycosylation patterns. But why is that important for cancer? Because we know that cancer transformation is the progression from benign to malignant. The cell gains a selective advantage for survival. And altered glycosylation is one such thing. It's a hallmark of cancer. Many glycosylation patterns are noted to change in cancer, like increase in fucosylation or glycan change branching and truncation, like on the left. But these changes are selective advantages that help the cancer cells overcome various hurdles and reach metastasis, like overcoming cell-cell interaction or escaping immune surveillance, modifying the cell matrix interactions. And these changes but the thing to remember is that these altered glycosylation is not all or none. It's the ratio of the glycans that matters. So how to target these glycosylation changes? Majorly, there are two tools. First is anti-glycan antibodies, which is pretty self-explanatory. And the other is lectins, which are carbohydrate binding proteins with high selectivity. But the reality of these tools in action is a little different. Firstly, if anti-glycan antibodies can offer higher affinity than lectins, but latest literature suggests that anti-glycan antibodies barely cover 5% of all unique glycan epitopes. They are also quite difficult to generate due to lack of rigidity of the glycan structure themselves, and hence they are in poor quality. Lectins, on the other hand, have a very broad glycan coverage and with very specific targets. It's like a lock and key. If we consider the lectin a lock, then the glycan is the key. That's their merit. But their affinities are lower than antibodies. That is in micromolar ranges, when antibodies can be in mid-nanomolar ranges, which is something that can be improved. So we can target the cancer-associated glycan changes by using lectins. And there is literature supporting it. Glycovariants of tumor marker AFP were identified in liver cancer based on affinity to LCA-lectin through electrophoresis, and that method is still used today. There are lectin microarrays used to identify mucinous cancers, and then lectin-based immunoassays, which can differentiate between CA125, which is an ovarian cancer marker, and they can distinguish it from benign diseases of the benign gynecological diseases just based on lectin binding alone, even when baseline level of the tumor marker CA125 is similar in range. Pancreatic cancer is one of the cancers we are focusing on. It's one of the most lethal malignancies. Its incidence ranks at 12th most common, which doesn't sound like a lot, but the mortality stands at 7th most common worldwide. That is a big jump. Being diagnosed with pancreatic cancer is like a death sentence because most people usually have 6 months at the time of diagnosis. With vague symptoms like jaundice and weight loss, they tend to delay diagnosis. And patients who are at high risk, they have it worse because they have to be screened frequently. And when knowing that the marker is not specific enough, that is terrible. Currently, the lead marker for pancreatic cancer is CA19-9, which is a carbohydrate epitope that's found on glycoproteins. And the test measures total tumor marker content only and doesn't consider the underlying protein. But this marker itself has many demerits, as we can see. It's commonly elevated in many gastric disorders. Some lung conditions also show elevated expression of it. That's the lack of specificity of the tumor marker right there. And for people at high risk, this leads them to sometimes get pancreaticoduodenectomy, which is the surgical removal of the pancreas, because the marker was elevated, but later it turns out that the marker was elevated due to a benign condition. Then additionally, 5 to 10% of global population does not express CA19-9. It's a variation. And those people will always test as negative, even if they have cancer. So not only that diagnosis can get routinely delayed, but some people can never get correct diagnosis because of it. And although there are many other markers in research, so far, none of the blood-based protein markers outperform CA19-9, which this paper summarizes very well. And it leaves pancreatic cancer without any early or good diagnostic markers. Which brings us to our focus, how to improve the existing tumor markers with the help of lectins. Firstly, we want to improve the functional avidity of the lectins by conjugating them to nanoparticles. Then we want to target glycovariants of tumor-associated antigens by tracing them with said nanoparticles. And specifically speaking, we want to detect multiple glycans on the same tumor marker, which has never been done before, to find lectins that identify malignant samples. This way, both the protein and glycan contribute to improving the specificity. So my work started with establishing a lectin nanoparticle library. We can see there are various lectins and they have different carbohydrate targets, different origins, everything. And we decided to use lanthanide doped nanoparticles for improving signal and the functional avidity. So based on EDC NHS chemistry, each lectin was conjugated to nanoparticles. And it means basically that there are many lectin molecules on the surface of a single nanoparticle. The key was how to keep these particles stable because every lectin is unique. So the pH, stability, and the buffer conditions need to be standardized for each of these lectins individually. Even the molar ratio of EDC and NHS needs to be adjusted. Otherwise, it can lead to precipitation. And the surface chemistry varies from lectin to lectin. So quality check is very important for each conjugation. And to do that, we chose to test these nanoparticles against specific glycans that are expressed in cancer. Again, if we consider lectins as a lock, then the glycans are the key, and lectins will only bind if they are functionally sound. So if the conjugation conditions are not suitable, then there won't be any binding. This is how we performed it. Basically, we immobilized biotinylated glycans and then traced them with lectin conjugated nanoparticles. This is representative data of one such experiment that we performed, where we can see that lectin-1, the blue dot, is binding to 3 different Lewis antigens because they share a common glycan that the lectin-1 binds to. But we can also see that when conjugation condition is not suitable, there is no binding. For example, in case of lectin number 13. Then we moved on to testing these lectins on tumor antigens themselves. For that, we chose to do it with spent media of different cancer cell lines. The spent media is known to contain the tumor markers because they are shed or secreted into the cancer cell surroundings. This is a representative data again showing one such test where we chose the tumor marker CA19-9 specifically, which is for pancreatic cancer. First, we tested for CA19-9 levels in the spent media, just like the test is usually done, detecting the glycan from both sides. And we identified that spent media from 2 cancer cell lines was found to contain the CA19-9 tumor marker. Cell line 2 is of pancreatic origin and cell line 3 is of colonic origin. We can see that 2 different sources of CA19-9 are right there. Now we wanted to see how these lectins are able to differentiate between CA19-9 from different sources as close to real scenario as possible. So we spiked serum with the CA19-9 tumor marker from the spent media. But we normalized the levels of CA19-9 tumor marker first, which we can see over there. And then in that serum, the CA19-9 tumor marker was captured from one side, as we can see in the image on the right. And then we traced the CA19-9 carrying molecules with lectin nanoparticles. We identified 3 specific lectins in this case. Firstly, lectin 1, it showed higher binding to CA19-9 carrier from pancreatic origin. And lectin 2 also did the same, but the levels are very low. And this clearly shows that lectins can differentiate between same tumor marker from different sources, which is what we want them to do. And then there was another wonderful finding. Lectin number 8 showed binding to CA19-9 carriers from both of these sources equally. Remember that the levels of these markers was kept similar. And this indicates that some lectin may also detect glycans that are pan-cancer in nature. And that points to a wider range of possible applications of the concept than just one cancer. My work is still going on. It includes testing the glycan patterns on other known tumor antigens like CEA, CA125, and CA15-3, all glycoprotein antigens. And my plan for future work includes the identification of such patterns on other cancer-associated glycoproteins, not just tumor markers. And then we can move on to validating the findings in clinical serum samples and in controls. I would like to thank my supervisor, Dr. Gaurav, our collaborators at AIMS, Dr. Pramod Garg, my RAC members, my family, my friends and lab mates for their support, and for my funding agency, DBT. Without all of them, this would not be possible. Thank you.
As we draw curtains on this stimulating session, we are reminded of the incredible depth and diversity of science happening within and around THSTI. A heartfelt thank you to all our speakers for sharing not just their data, but their passion and purpose. May this exchange spark new collaborations, inspire fresh ideas, and remind us of why we do what we do. With that, we conclude the scientific colloquium, and we thank you all for your rapt attention and scientific curiosity.
We are deeply delighted to have Dr. Alka Sharma with us here today. Dr. Sharma is Senior Advisor and Scientist H at the Department of Biotechnology, Government of India. And she is former Managing Director of BIRAC. She has played a key role in advancing biotech R&D, shaping national policies, and fostering public-private partnerships across the country. Currently, she heads the Biomanufacturing Directorate under the BioE3 policies, aligned with the national priorities like green growth and Atmanirbhar Bharat. A recipient of CSIR Technology Award, Dr. Sharma also trained at NIH, USA in an extramural research management grant. Given that Dr. Sharma has nurtured THSTI since its conception, no one can better describe THSTI's 16-year-old journey through the lens of DBT.
Thank you. Thank you for nice introduction, nice words. So very good morning to all of you. And it's my pleasure to be here with all of you today on a special occasion, 16th Foundation Day of THSTI. So I tried to put together, but I can see veteran here, Dr. T.S. Rao, Dr. Shinjini, and many, the old faculties. This I have just prepared, keeping in mind the research students, young faculties, and also some of the participants who are here just to hear about THSTI activities. So the title has been given to me by THSTI, so I tried to fit the things actually what has been done so far and from where it started and where currently we stand and what is the way forward. So first of all, happy 16th Foundation Day to THSTI. So this is the time to, to celebrate this and the way actually I said that Dr. T.S. Rao is here and Dr. Shinjini, myself and Mr. Santu and many more actually, they are associated with THSTI, I think right from the beginning, from day one when Dr. M.K. Bhan, Professor M.K. Bhan, who was actually under his leadership, who was by training was a pediatrician, but many of you, all of you know that he was former secretary also of Department of Biotechnology. So foundation stone was laid down in this campus. But before that, this was in this initially operated from an interim facility in Gurgaon, and then it was shifted to this campus in 2014. So interim facility was started in 2009, and then now it has been transitioned into a model organization for clinical and translational research in India. Gradually, the THSTI has emerged as a premium translational institute of international repute and now bringing fruits of biomedical innovations to the public. In this slide, you can see the gradual and strategic growth of THSTI aligned with the institute's mandate for translational research. So initially, THSTI had focused on basic research in disease biology to build a deeper understanding of the underlying mechanism of key health challenges. Building upon its early discoveries, THSTI strategically expanded into preclinical research, leveraging cell-based assays and animal models to study disease, the pathogenesis and vaccine efficacy and so on. So the THSTI then progressed into clinical research, a core component of its translational mission, with the establishment of CDSA and partnership with clinical institutions across the country and also outside the country. THSTI focused on designing and managing clinical trials, and during COVID also THSTI played a key role in biospecimen collection, analysis, enhancing national readiness for public health challenges. Similarly, you all know that about major programs on cohort studies of Garbani, Translational Research Program, TRP, Biodesign, Biotech Cluster, and also recently initiated Biomanufacturing Program under Precision Biotherapeutics. So moving ahead, THSTI also in the process of establishing Bio-Innovation Park under Bio3 policy for fostering high-performance biomanufacturing. Dr. Manish Diwan is here, who is driving this activity, and it has gone to the advanced level. So this actually at a glance I wanted to share with all of you that these are the major activities which have been taken in the last 16 years, but there are many more that are offshoot of these major activities. So the THSTI has beautifully integrated basic preclinical and clinical research within biotech ecosystem, supported by immunology core facility, vaccine design and development facility, small animal facility, challenge models for development of vaccines, biorepository, and CEPI centralized bioassay laboratory. So now THSTI has now matured into a nationally important research institute capable of driving innovation across the entire biomedical value chain. So now onwards, I will very quickly run through the slides for the programs which are now has taken the major the role in promoting translational research. So as you can see that I mentioned in my initial first slide, the CDSA was established and there was a dream program and center of Professor M.K. Bhan, and it's a first of its kind center which has been started in THSTI on an experiment basis, because this is something that was required, and we know today also, and that it was done more than a decade but earlier. But now also, if you really see, one CDSA is not going to cater the need of the requirements which we have in the country. So this is something that we need to expand this further. And also this, the mandate, you can just see that this is something that CDSA is supporting and nurturing clinical research capacity in India for clinical product development, and this includes clinical portfolio management, clinical sciences, and also a strong component of training programs. And this was very, very helpful during that COVID also, when PACT was conducted in partnership with BIREC and DBT. And at that time, 2,400 participants from across the globe, they were trained in clinical trial management. And this is something actually very important component of CDSA that you can see in the slide. Similarly, THSTI is very strong, has a very strong partner. This is one of the partners of NCR Biotech Science Cluster, and driving translational activities along with other stakeholders through interinstitutional and cross-disciplinary linkages for science, technology, and education. And the focus of THSTI was actually that how to bring together all stakeholders, and all major facilities were supported by Department of Biotechnology at that time. And it was a sort of the society, but later on it is now a core part of this whole campus, and THSTI, RCV, and other BRIC institutions are also partner of this cluster. And this you can just see BSL3 facility, ATPC, incubators, Office of Connectivity, Animal Facility. So that was the vision of Professor M.K. Bhan, because today when we talk about all these facilities, and people are now talking across the globe and also in the country, but that was the vision of our leader that time, and he thought that that is the requirement of our country to take the translational research to the logical conclusion. And that is something that we all work together from DBT, THSTI team, another BRIC institution. Now we call them BRIC, Biotechnology Research Innovation Council institution, but at that time they were autonomous institutes of DBT. So this is something very, very important to know for all of you who actually don't know about this. So again, this is something that THSTI has played a very important role in establishing and driving biodesign program through Center for Biodesign and In Vitro Diagnostics. Overall aim is to develop this in vitro diagnostics, and also that to develop medtech innovators to foster and promote medical technology innovation in India. And at that time, when we are talking about this is 2007, there was no innovation ecosystem in the country, and there was no BIREC also at that time. So this is something that Professor Bhan visited Stanford University, and he saw that there is something, the program like biodesign, and we have to bring together all stakeholders, medicos, engineers, product designers, and also MBAs and all to manage the program. And he tried to develop the same kind of maybe the something some program which is suitable for our country, but mainly based on the concept of biodesign process. And we started, and then at that time there were only 4 centers were established by DBT, and THSTI was one of them. And since the focus was on in vitro diagnostics, so the partners were THSTI, ICGB, and AIMS, and the international partner was University of Turku. So what actually what are the outcomes? If you really see on the left side of this slide, then the number of innovators have been trained, technologies have been developed, startups have been created, products are already there in the market, biomedical training have been conducted. So what happened then, the fellows who were trained in this biodesign process, they attracted funding from national programs and also international Canada grand challenges, welcome trust and all, and they developed all these products. Basically it was end-to-end process, right from clinical immersion up to product development. And in the process, they also created and established the startups which were supported later on by BIREC. So BIREC came into existence in 2012. So by that time, the fellows, they all got matured and their prototype, alpha, beta prototypes were ready to get the funding from BIREC and Wellcome Trust and other agencies. So this is actually now you can just see the who all have involved, have actually carried out the various kind of experiments in THSTI. Today you can see the fruits of those. Translational Research Program, TRP, this is something that trying to bring together all the faculties, students, and also the scientists, and also from other institutions in partnership, how to develop translational research which is going to deliver something which is very, very important for the country and aligned with the national priorities. So the TRP, you can just see that the focus was on infection and immunology, diagnostics, maternal and child health, non-communicable diseases, and that outcomes are also now listed. But since I am in THSTI, and so you know much better than me, and you that's because most of the scientists here, they are the part of TRP. I'm not going to elaborate on the outcomes, but this is something that phase 1 has been completed, and now phase 2 also. So the leads, based on the leads available on phase 1, phase 2 has been developed. This is also very important and very, very key program of THSTI. So going forward, so this everybody knows about Garbhani, and this is one of the largest hospital-based pregnancy cohort in South Asia. And the focus is on how to generate data which are specific to our country, and then based on that, some contribution, significant contribution can be made to the policy makers, and also for our own country contribution, that they can have a reliable data and also reliable policy which is going to have some kind of implications, that the positive implication that critical risk factor will guide prioritization of interventions, risk stratification models permit targeted delivery, because the number of preterm birth and SGA that Dr. Singh is here, she actually started this, and then later on with the this partnership with other stakeholders within the country, outside the country, now this has been expanded further, and now many more programs have been developed based on this foundational thing that was started long back, and now everybody is aware of this program. Just take the name Garbhani and they can connect it well with the outcomes of this. NCEPI, this is something was started, and this is something that was the first of its kind in the country. Dr. Vijay Raghavan was the secretary at that time in DBT, and he was the founder member of CEPI. At that time, it was started by Norway, and the India was the founder member. Even it still is there, but we are not contributing in terms of that. We are just supporting to our industries for developing vaccines and other products. But now they actually have more partners, but we are the founder member of CEPI. So what we did actually aligned with CEPI concept, we have established India-centric CEPI, that is NCEPI, and the THSTI is the major beneficiary to have funds from CEPI and also NCEPI, and BIREC, the PMU has been established in BIREC, and the facility BSL3 and also the translational laboratories which have been established in THSTI, that is from support from CEPI. And that you can also see the achievements under CEPI. If you really talk about vaccine, so mRNA vaccine got emergency authorization which was developed under this. Chikungunya vaccine is under phase 1, phase 2 clinical trial in 7 countries. India is one of them. And both this beta coronavirus and monkeypox, they are advanced in the preclinical stage. Shared infrastructure facility, I have already said that. But the important thing I would like to say that whatever this CEPI centralized network lab which has been established with the support from the DBT and also the some know-how from the CEPI, now is one of the this is the first of its kind lab in India, and they have recognized globally as one of the CEPI's lab and the BioSA lab. And also, as I said, that the consultation with other interministerial, mainly ICMR, and also the PACT PACT that I actually talked about this earlier. So there are many countries that participated, and 2,400 participants, they actually got benefited from this. So that was under CEPI program. And this is actually just wanted to highlight that the CEPI lab and bioassay lab in the country that is actually in this is housed in THSTI, is one of the the lab which has been recognized by CEPI. Coming to Bio3 policy, that you all are aware that, and that this has been approved by the Union Cabinet and under the Honourable chairmanship of Honourable Prime Minister. And this is, there is a commitment to foster an advanced future that is more sustainable, innovative, and responsive to global challenges, and also for advancing national goals through bio-based innovation to build a robust and sustainable bioeconomy. So I'm just going to tell you about, these are the 6 thematic areas. I'm not going to read them, but precision biotherapeutics, which is third one, this is related to THSTI. So, and also the lower portion of this, lower part of the slide, that's the bioenablers. So BioFoundry also is now THSTI coming forward to establish BioFoundry under this initiative. So precision biotherapeutics, all 3 sub-verticals, cell and gene therapy, monoclonal antibodies, mRNA therapeutics, THSTI are involved in this in a big way. And a SWOT analysis have been carried out to prepare a strategic roadmap. Many of the faculties and also the scientists, they are actually involved in contributing towards this formulation of a strategic roadmap based on a national, international scenario, existing strengths and opportunities, and also gaps and challenges under 3 categories: R&D, bridging the gap, and pilot and pre-commercial manufacturing. Regulatory, to address the regulatory issues under these 3, that is also very important. Cell and gene therapy regulatory conclave has been organized with all the stakeholders. Other 2, we are now in the pipeline to do that, and also establish public partnership is very, very important, and international collaboration. That is nothing to do with the cell and gene therapy and all these 3 subsectors, but I just wanted to share that DBT has signed a MOU with National Science Foundation USA, and that will cover some of the part of omics on these verticals under this. So as I said, that the implementation is mainly under 3 categories, that is the research and integrated discovery and integrated research network, bridging the gap, that is scaling up, that's the crux of this whole initiative. This is very, very important for the country, and also setting up a biofoundry and biomanufacturing hub and bio-AI hub across the country. So basically, the overall aim, what we are envisaging is that to position our country amongst the global leader in biomanufacturing by creating a strong innovation ecosystem, by promoting and facilitating scaling up pilot and pre-commercial manufacturing, leveraging and mobilizing finances for, especially for public-private partnership, that is very, very important. And also there is a need to have a coordinated framework for the regulation under each vertical, including precision biotherapeutics, which I showed that. And overall aim is how to integrate all these things in the bioeconomy. So as I mentioned that in bioenabler, that THSTI has submitted a proposal, and now this has been approved and this has been implemented. Basically, the funds have been released, and now they are going to set up biofoundry and THSTI. And this is very, very important because the subsectors which I mentioned about precision biotherapeutics, so that is now in some manner it is going to be very, very useful for scaling up, and also the pilot manufacturing with other stakeholders in public-private partnership. So overall aim is to create infrastructure platform for product development, process innovation, testing, and scaling of biotherapeutics. So basically, the role of THSTI is very, very important, and it has shown in the last 16 years also, right from the beginning, and how to create biovision for Viksit Bharat. And without THSTI, we can't think about it, because the kind of contribution that THSTI is making for this particular, the vision for building a more prosperous, equitable, and sustainable development of current and future generation, and in the biopharma and other bioindustrial, bioenergy, bioservices. These 4-5 components you can see here, THSTI is playing a major role and contributing towards this. And this is my last slide. So basically, what I presented so far, this has been done. But this is something, it is under process, as I mentioned in my first slide, that this is now the DBT under the leadership and guidance of Dr. Rajesh S. Gokhale, Secretary DBT. This is now being discussed at various level, and this is called IBRIC Plus cluster, and this brings together institutions, industry, and also for creating infrastructure and proof of concept to pilot scale facilities, early stage manufacturing facilities, and capacity to generate data for regulatory submission. So basically, the outcome you can just see at the bottom of this slide, that is high value bio-innovation through industry academia collaboration, talent nucleation, promoting biomanufacturing jobs and boosting bioeconomy. And this is the full form of Bio3 policy, biotechnology for economy, environment and employment. And this is directly related to this. And we are hoping that in the next Foundation Day, we will be here. I think this we will talk more about this, and by that time, some part of implementation will be taken place. And this is the aerial view of that where you are sitting, and you all know about it. Thank you very much, and thank you for the organization giving me this opportunity to share with you all about it. Thank you very much.
Thank you, ma'am, for invigorating the spirit of our institution with your words. Ladies and gentlemen, one of the most awaited moments of today's celebration has arrived. The Foundation Day Lecture, a tradition that brings us the wisdom and perspective of a luminary in science and public health. It is our distinct honor and privilege to invite a visionary whose contributions have left a deep imprint on the global biomedical research landscape. Professor Anil Kaul, a leading expert in drug discovery and R&D, is the former Vice President and Head of Global Public Health Discovery and Partnerships, Johnson & Johnson. He also serves as professor at the London School of Hygiene and Tropical Medicine, where he leads a lab focused on TB translational research. Professor Kaul is best known for discovering and developing bedaquiline, the first approved drug in over 45 years for drug-resistant TB, which is now on WHO's list of essential medicines. For his breakthrough, he received prestigious awards such as the Sun Pharma Research Award in 2017, American Chemical Society, its Heroes of Chemistry Award in 2020, and the prestigious Prix Galien France Prize. He is also named one of India's top 20 bioscience innovators by BioVoice News. Please join me in warmly welcoming Professor Anil Kaul to the stage to deliver the 16th Foundation Day lecture titled From Bedaquiline to J&J 6640, New Modalities in Treatment of Tuberculosis, What the Future of TB Holds.
Can you hear me? Yes. So, good morning, everybody. It's such a pleasure to be at this fantastic event. And it's also very pleasure because I have seen THSTI grow and being built as an institution. Probably you might have known or you might have seen that we are sitting in MK Ban Auditorium. Professor Ban was an amazing visionary. I think tribute goes to him before I start anything. I have kind of met him, I have seen him, and it has been such a pleasure to see a visionary who built this institution right from the scratch. And I am able to speak today in this particular auditorium, which is aptly named after him. So I think before I go and talk, I want to thank the organizers, the Singh. I have been at many institutions across India, across globe, and I can tell you that this initiative of industry meeting academia and leveraging that connect is something which is very, very valuable. I come from both the worlds. I have seen the industry. I've been with J&J for almost 20 years. I'm associated with the Indian institutions. I have been associated with the London School. So I know how important it is for any translational science to know the language and to talk to each other. Because as scientists, we tend to be absorbed too much in our labs. But as we develop programs which are translational in nature, we need everybody to come together. The teamwork is the key. So thank you very much. Thanks to organizers. Thanks Manish and all the Sinchen team and everybody who has been involved here. So thank you for bringing not only me, but rest of the industry folks. I see a lot of the industry people here for bringing us here in this fantastic THSTI institution. So before I go into the main deck, I also was very, very much impressed by the amazing science which the students present this morning. We didn't have a chance to talk to you or question you, but I would love to talk to all the students after we finish this particular day and interact a little bit more under the depth of the science you have, the microbiomes, the pre-eclampsia, and all things which you were talking. Really, really impressed by the quality and dedication of the science happening in this particular institution. And I can tell you, institutions are built not by auditoriums, they are built by people. They are built by the talent. And the talent is the most valuable thing. Sometimes we underappreciate the talent. That's our, in the Indian institutions, we need to bring forward the talent and appreciate the good science and the good people which bring the best in these institutions. So thank you once again. So let me go to the main slide. So I will be talking today about Bedeclin. So a lot of you have heard about the story. So I will briefly mention it for the people who are new to the audience. But I will mostly talk about the new regimens and the future of TB treatment. So last thing, few slides, I will talk about my aspiration of world without TB. I think it's our national aspiration now. Modi ji says it everywhere. I don't know whether it registers in you, but at least it registers in people like me that, yeah, we need to do something to this disease which has been creating havoc in this particular country. We are 30% of global TB burden in this particular country. And we talk about so many big things, but we still have not addressed a disease which is almost, thousands of years old. So I didn't want to hide myself next to this podium. I don't have a pointer which can move the slides very fast because I wanted to talk to you. I don't want to lecture you. I wanted to talk to you. I wanted to see your expressions, whether I'm connecting to you or not. But if you look at TB burden, I don't need a slide on this. Everybody in this audience knows it. But I just wanted to reiterate the fact that we have almost 1.5 million people dying of TB every year. And 30% of those deaths happen in India. So, and then the biggest challenge beyond the normal TB is the drug resistance, is the emerging resistance to so many different drugs. So if you look at drug resistance, what does it mean? I think I try to show this slide everywhere. So you see that this is normal TB, this is MDR TB, this is pre-XDR TB, and this is XDR TB. So those of you who are TB researchers or the clinicians understand it very well. But those who are new to the field, they understand that this is basically a bacterium which is resistant to rifampicin, isoniazid, fluoroquinolones, as well as all kind of injectables. So everything, this bacterium, you give this patient any kind of drug, they don't respond. So most of the people who had XDR TB or MDR TB, the mortality in XDR TB, the mortality was almost 80%. So it is worse than a cancer cancer patient. So I was lucky enough because I have been in this field for very, very long. And I had a chance to visit and this particular guy, Professor Srivastava, who was the head of TB division in Banaras. So it was 2019, and he gave me when I went to his place, he warmly welcomed me because at that time, at that moment, we had, Priya is here with me, we had launch of Bedeclin in India, and he was very happy. I didn't understand why exactly is he so happy. He hugged me, "Aaja gale milte hain," and all that stuff because we had never seen each other. So then he told me this story of this 24-year-old man who had an MDR TB for 1.5 years. This was his thing. He was on a WHO conventional MDR regimen. The challenge was his family history. Father and brother died while treatment on XDR TB. Mother diagnosed recently with XDR TB. And the younger brother was the only one in the family who didn't have XDR TB. And the whole family was slowly and slowly dying. That's the challenge of TB in this country. If you have an XDR TB, you can be guaranteed that no family is safe from this particular disease. So that's why we need new drugs. We need to save these patients. These are our fellow countrymen. We are a nation of 1.5 billion with such a burden. We need new drugs. But drug discovery is a lot of serendipity. If I tell you that tomorrow we will have a drug, that will be challenging, but it needs a lot of serendipity. But apart from serendipity, what it needs is the time. It takes 12 to 15 years to develop a new drug. It costs a lot of, it's almost 1.5 billion on an average. What it takes for oncology, I'm giving you oncology drugs or the neuroscience, that it takes. Like TB, I just give one example. It takes MSF almost $40 million to do a single phase 2B trial. And of course, the chances of success are very limited. But I'm a very optimistic guy. I drive myself by sheer optimism sometimes. Sometimes it's misplaced. It's not always the right thing to do. But yeah, you can't change a person. So the optimism says that whenever something important whenever something is important enough, you do it even if the odds are not in your favor. So it's a quote from a very famous guy. So, but I really believe in this kind of approach. So coming to bedaquiline, this was the drug, TMC-207 or 207-910, diaryl completely new chemical class, completely new chemistry. We showed it for the first time in that it blocks the ATP synthase, which is shown here with an IC50 of 2.5 nanomolar. So basically what the drug does, it shuts down the power supply of TB bacterium. So that's what the whole thing does. It's a completely new mechanism of action with which we had discovered. So this is for the students here, this slide. I just specifically made it for you because when we were discovering bedaquiline, we didn't know exactly what it did. We had resistant mutations on the ATP synthase, but then we said that it's not really conclusive. So at that time, I was still working in the lab. So we created this molecule with this amino group and we connected it to the linker and connected it to the cephros, and we had the control resin. At that time, everybody said this experiment is going to fail because the drug is so lipophilic, it will bind to the resin, everything will come down, you will not have any, we call it affinity resin, everybody knows it now. So, but to my surprise, when we did a silver stain on this, there were only 2 bands. I said that this experiment has never worked. So it's non-specific, nothing has come out. ATP synthase was an oligomeric of 8 or 9 protein, only 2 proteins here. But so we were about to throw this gel, about to just discard it, that this has not. And somebody said, why don't you do an MS of these 2 particular bands? And luckily at that time, MS was not easily available. We had to give it out. So what MS said that it was alpha subunit and beta subunit of ATP synthase. It was a big huge surprise because this is where we had localized the resistant mutants to this particular drug. So which was very, very surprising. So the lesson learned is basically things that seem obvious to fail in all the experiments what we do might work. So the key is try, keep on trying. So coming back to the clinical data, some of you are clinicians here. I put this slide. Basically, this is a New England Journal of Medicine study in 2020. We showed that these are populations with XDR TB, which I told you that had no cure. They needed drugs for 24 weeks plus daily injections for 6 months. Every day these patients had to go. So we use this population, XDR TB population, MDR TB population. This is overall ITT population. And you can see the cure rates of bedaquiline containing regimens, almost 90 to 92%. Before the advent of bedaquiline, this cure rate in XDR TB was less than 30%. maximum 30 or 40%, 30% maximum in best hospitals across the world. But now you can see what it changed. It changed the dynamics of the TB in across the whole globe. It changed it from an injectable regimen to a simply oral regimen with a success cure rate of almost 90%. That's why it became one of the cornerstones of MDR TB treatment across the world. So when we measure it, first drug to be developed with a new mechanism of action in 50 years, 1 million doses have been given to patients across the world right now. So that's the success of this particular drug. 5 million infections using bedaquiline have been prevented. 170 countries have access to this. I think somebody said yesterday, which I completely agree when we were having the panel discussion, that it's not only the medicines you discover or you do clinical studies, it's the access. How do you bring a medicine across 170 countries? And we are talking about CAR-T. How do you bring CAR-T to the every cancer patient in this country? So you won't, the THSTI will not only suffice. How do you bring it to some, we all come from different places. I come from a very small village. Where exactly, how does the patient there and have access to the cartridge? So the access is very, very important. So the drug was 73 countries and bedaquiline has been approved, all oral. It's very important that injectables were removed from the treatment regimen because of the bedaquiline. It's on WHO's list of essential medication for adults as well as children. Tackling pediatric TB. This is one of my passion because 1 million kids have TB every year. And what we were able to do, we were able to do discover and develop a pediatric formulation, dispersible formulation for kids. And that got approved by US FDA in 2020. And it's also on the list of essential medicines for children to be used. What was very important is that this report in Times of India in 2024, where we showed that 4 kids with drug-resistant TB were cured who had XDR TB. Imagine a child as old as 4 months had XDR TB. And when they gave bedaquiline, they eventually got cured. Means at least they, as per the report, they were saved. So now we come to the next part of my talk, which is the basically new regimens. But before I talk about new regimens, I want to give you a general outline of what we call the bad. We are doing less with exponentially more. So a lot of government, a lot of across the world, NIH or US government, they spend a lot of money in R&D research. But the people are questioning, is research in these days giving you the value? Are we productive enough as institutions, as countries, in all the dollars we spend upon the research? So you can see that science is getting less bang for its bucks. There are a lot of reports, a lot of analytics which are happening that whether the research is giving you the quality. So this is what is called Irum's law. I think this is something very new. It was also very new for me. So Irum's law, don't go to your managers and say that Irum's law hai to hum research funding itna useful nahi hai, but this is just for the science of it. So the Irum's law says that the exponential drop of productivity, and you can see that the drop of productivity in creating new molecules, it's mostly from the pharmaceutical industry perspective, that between 1940 and 2020, you had an exponential drop in the number of NMEs per $1 billion spent. So now for every $1 billion spent, we have only 0.7% chances of getting an NME. So that is what is Irum's law. The failure is norm. That's what we were told, that the failure is norm. The median cost of developing a drug is almost 1.1 billion. And the trend is reversing because we have now genomics, we have molecular and personalized medicine coming through. So that's something which is very important to understand, that now we understand biology much better than what we used to do. And because of that understanding of the genomics and molecular and personal diagnostics, there's a reversal happening right now in the last 5 years. So now you can see this red dot here that there is more molecules coming out because of this particular thing. So when we talk about TB, the same thing is happening in the TB R&D. So what is happening in the TB is that we understand TB pathogenesis much better than what we used to understand 20 years ago. So we understand that why TB is replicative as well as non-replicative, dormant, that it goes inside the patient, it stays there. Why it can be intracellular, at the same time it can be extracellular. Why, what kind of a nutrient availability is needed and what kind of a oxygen concentration is needed. So the dynamics of TB biology has expanded. The way it has expanded in the cancer, where we understand the genomic studies have helped us in defining cancer much better, making it more personalized, like CAR-T. CAR-T is exactly a personalized cancer treatment. And here we also, once again, with these kind of understandings, we understand TB biology better. So that's why the Eroom's law, which I showed in this particular slide, may not be applicable when it comes to the new therapeutics for TB or any other infectious disease pipeline. So this is the TB therapeutic pipeline. You can see that I just wanted to show you that this is the pipeline where we have the major drugs. And this is the diarylquinoline class. This is the cytochrome B class. This is the DPRE1, oxazolidinone. So different classes. So the pipeline is enriching and moving forward. So WHO says, what do we need in a TB new drug? So WHO recommends that in a new regimen, now I'm not talking of bedaquiline-based regimens, I'm talking about new regimen, we need 3 to 4 months of treatment. So right now that regimen says we need 6 to 9 months. Now WHO says that the next wave of innovation in TB has to be 3 to 4 months. It can be a fixed dose combination, that's very important. And we have to remove one of the drugs which is currently used in MDR, TB, linezolid, which gives you a lot of side effects. Especially in the Indian population, we have seen linezolid as a major challenge because it induces myelosuppression in patients. So we wanted to remove that thing. So as I talked about Eroom's law, the loss of productivity, but better understanding of TB disease pathogenesis, but there are other tools which are coming into the operations right now. One is, everybody has heard about it, AI. The AI functioning in drug discovery is really revolutionizing the whole field. And I personally believe that this is going to transform and democratize the neglected disease R&D space. So within the AI, just give you a very simple example. One of the things which I am bullish about is generative chemistry. So the generative, Akhil is here from, I know him from Origin. They have a platform which is very interesting in terms of using this platform because while you need a simple X-ray structure and you can, without screening millions of compounds, you can define the binders or inhibitors, either a small molecule as well as antibodies at times for this particular thing. You can do more augmented molecular design. And then in terms of the TOX and other platforms, we have seen biosignature platforms. So there are different tools at different processes during the R&D development, which is AI driven. And I feel that we as a nation should be putting more effort in the AI-based drug discovery and clinical development efforts. And that's where we will land up with a new TB drug. So when we started with this particular drug discovery program, you know, as I talked earlier about bedaquiline, so this was the target of the bedaquiline. So what we thought at that time, maybe we need drugs which synergize with bedaquiline. So then we said, okay, let's look at different drugs. We looked at this particular molecule. This is cytochrome BC. We looked at this one. We looked at several central carbon metabolisms. And the idea was that because this is a chain, this is like a cycle chain. So if you block this one, but also block this one, maybe that you develop a synergistic drug combination, which can reduce the treatment duration needed for TB. That was the whole idea. So today I will talk about purine biosynthesis, the latest data what we have. So purines are basically, this is the biosynthetic pathway of the purine, and they are basically involved in synthesis of nucleic acids, RNA, DNA, and other nucleic, very essential molecules for the life. So that's what the purine biosynthesis is all about. So this is what we discovered. We discovered this molecule called JNJ6640. As a very novel purine inhibitor. The initial molecule was 380 nanomolar, quite potent molecule, but chemists have a magic in them. I always say that the best biologists are the chemists turned into biologists because they understand the biology much better than the biologist. So what they do, they convert this particular molecule almost 40-fold and improve the potency of this molecule to nanomolar. And you can see that it was very, very potent in terms of killing TB bacterium out there. So of course, we wanted to understand what does it do basically. This molecule, we went to raise the resistant lines, and we found that the resistance was in the PureF, the protein involved in the purine biosynthesis, and we mapped it here, and we developed this particular molecule in the binding site. The question is always, humans have purine. We all have DNA, RNA as building blocks. And how does this particular molecule, will it kill the humans? Because this is so conserved pathways. So we looked at different cell lines, we looked at different human purine analogs, inhibitors, but you can see that JNJ6640 is green. So whenever you see on a slide something green, imagine that it's okay. So this is that it was very clear. And then we developed a very nice assay looking at the human PureF. This is the human PureF versus mycobacterial PureF. So it was very, very selective for bacterial enzyme and less selective for human. So these basically purine biosynthesis has been used as a target for cancer therapy in humans. That if you block the purines in cancer patients, it can basically block the cancer replication. The challenge is always with purines. This gets a little bit technical. Sorry for that. That the purines can be also, bacteria can get the purines not by synthesizing itself, but from the lungs of the patients. So they take these purine building blocks and they can salvage basically. Then you don't need your insert. So we were asked the question that, okay, yeah, if you put the exogenous purines, then what really happens? And you can see that if you put the hypoxanthine, which is also an nucleobase, you can rescue it. So that was the biggest challenge, that will in humans the purines be rescued? So yeah, sorry. So what we did a metabolome study in lungs. So we went to the patients. I think this is the beauty of this particular research work, I'm saying it, because we went to the human lungs infected with or uninfected with TB and did the metabolomics for the first time. And you can see that the level of the purines, what we measured in the human lungs versus non-infected lungs, with mouse lungs and with mouse non-infected lungs, that they were nearly that we created a data set where we said that no, the exogenous purines will not be able to complement inhibition of PureF. So I won't go into the details. And then finally, the key is this one, this slide. In an MDR TB patient, the current regimen is BPAL, bedaquiline, protonimide, and linezolid. And I told you that linezolid is one of the worst drug for these TB patients. We are trying to replace linezolid. So when we replaced linezolid with 6640, you could see that we had much better activity in killing TB bacterium as compared to the controls here. And when we went to the mouse, we could easily replace linezolid with a very optimized formulation of 6640. So that means that this drug has the potential to replace linezolid in the future regimen and can eventually have better side effect profile and may also reduce the treatment duration. So finally, the last part is aspiration of world without TB. As I said, that what we need in a TB is the drugs which are bactericidal. We need to aim for a regimen which can be less than 3 months. We need to have oral regimen. That's very important that injectable drugs are not part of the regimen. They need to be compliant with the HIV treatment. We need to have a universal regimen. Imagine a patient coming in a doctor's clinic. So we don't need to do a DST, the susceptibility testing and all that. We can have a patient and directly put him on a regimen, which is basically pan-TB. And then we need, of course, better compliance. So my hope is that we have a TB regimen, which is a single pill, a fixed dose combination, a single pill for treatment of TB. How do we do it? I think we do it through different pillars. So pillar one is basically patient finding, identifying missing millions. You can imagine there are missing millions of patients which go undiagnosed TB. We need to expand the access. As I said, once again, the access is very important to the MDR TB medicines and accelerate the R&D. That's also, these are the three pillars which I see as the core of our national policy for eliminating TB in this particular country. But there is signs of hope. I think this is very important for us to see as a nation that we are moving on a positive trend in terms of eliminating TB. If you look at the latest data from WHO, there is a decrease in the TB rates and related deaths in the recent years in India. So that's something all we should be proud about. So the TB incident rate dropped almost 17 to 18% in the last 10 years. And there is 18% reduction in mortality since 2015. So we are on a positive trajectory in terms of addressing the MDR TB issue in this particular country. What's also very important, because we are in a translational institute, I want to tell you that it's very important for us to connect to the doctors or physicians which are treating TB. So this was one of my recent visits in KGMC in Lucknow. And the key was interacting with the doctors out there. And she is a patient here who had a spinal TB. The extrapulmonary TB in India is such a huge issue. And you can see that, now beyond the pulmonary TB, we also need to address the extrapulmonary TB out there. So coming to the THSTI, where we stand, and Alka has already pointed out, THSTI has a fantastic, talent in terms of Center for Excellence in the TB research. They can do disease biology, they have medicinal chemistry, they have diagnostics, therapeutics, host-directed therapies, and they have fantastic in vivo BSL-3 facilities. And now they are coming with this potential human infection model and the MRC Center, and they have linkages to major organizations like CEPI, ICMR, as well as the industry. So that's why I feel very hopeful that THSTI can make a huge impact in future TB drug discovery, or as well as the vaccine research which is happening across the country. So these were some of our key players involved, both in the Bedeclin as well as the purine biosynthesis story I told you. This is, I always say, lab to the last mile. So we always have to remember this when we say of the translation, what is the strategy? I talked about it yesterday. What is the strategy of moving drugs from your lab into the last mile? So, and there you need a lot of partners. So these were the partners in the lab, and many hundreds of collaborators. These were partners during the course of the development of this, and the last mile in terms of the supply chain. The USAID, which is now closed, Trump closed it, unfortunately, but they were very important in bringing the medicine to more than 170 countries, the Global Fund. What's also important, we had an MTV Live. We collaborated with MTV in terms of educating the youth, the younger generation. Sometimes they don't understand what TB is. It means a lot of these new kids, millennials, I don't know what the current generation is called, Gen Z, yeah, sorry. So बहुत confusion in Gen Z. Gen Z से पहले क्या आता है? Huh? Gen X. So maybe I should say Gen X को पता नहीं है TB क्या है or where the disease is coming from. So we created, maybe you should have a look at it. And we got some Bollywood stars talking about MTV on TB in terms of and the impact and social stigma and all that stuff. So you can see about it. This is my last slide. It tells you the story. I try to show it as much as possible. This is a story where we went to South Africa to a XDR TB site, and we looked at the pharmacy of this particular hospital. And when we looked at these boxes, I asked them, what exactly are these boxes with the BL written on top of each box? They said it is Bedeclin. So this shows you the power of the lab to the basically the last mile, the patients. And the patients are basically, they can be anywhere located in the world. It could be any pharmacy in India, but it was happening somewhere in Africa. So the last in terms of, can we change the trajectory of TB in this country? I believe, yes, we can change the TB landscape. This is what we discovered as Bedeclin. But with the new molecules, with a very advanced pipeline, with the vaccine also, currently there's a huge phase 3 vaccine trial ongoing across the world. I believe that in my lifetime, I don't know Gen Z or Gen X, whatever you call, in their lifetime it will happen, but in my lifetime, this is surely going to happen. I may see a world without TB. And this is the final thing, acknowledgements. The most of the PUREF work was done by Richard Wall. This is a very small team. It doesn't involve chemistry, it's just the biology team, pharmacology and everything was done separately. And with that, I would like to stop here. And if there are any questions, happy to take. If there are no questions, I'm happy to do it afterwards. I'm here the whole evening, so catch me if you can. Yeah, Nishit. Yeah, means J&J has done tremendous for the world in terms of the TB research. And policy change, strategies change, new direction change, but there are always other emerging leaders in the field of TB from the industry perspective, which will fill the gap. So it's just a matter of people venturing it and taking the ownership. But I believe like there are a lot of other people who are doing excellent work, which goes beyond the J&J labs or London School labs, like THSTI. Means you guys are really, really good in TB research here. Because I think what we sometimes don't do is appreciate our own selves. So we lack that appreciation of institutional appreciation. And I can tell you that the facilities what you are creating and what you have are the world class. They match anywhere else in the world.
The mode of action of bedaquiline, and do you see any connection between the drug you discovered and developed, bedaquiline, and the equally popular, possibly not as crucial, proton pump inhibitors?
That's the smartest question I have heard since several days. Yes, and there's a history to it. Bedeclin came out of a screen which was done for Helicobacter pylori proton pump screen. We were looking at that time, gastric cancer was really on rise, and we were looking at those proton pump inhibitors, and out of that particular screen came. I don't know whether there is a direct connection or not, but history tells you that the way the drug was discovered was through a proton pump inhibitor screen.
Anil, great talk and great work. I was wondering, I had two questions in mind. One is in terms of, we talked a little bit yesterday on the frequency of mutations, generation. And I was wondering, would you want to use it as a last line? You did give me the answer, but I still want the audience to know that. And the other thing is that if you were to use it in the first line, what would be the frequency of it? Have you tested that in terms of the mutant generation if you were to use it in the first line?
Yeah, I think some of the people here know the data much better than me because they discuss this paper in their journal club. And I was apprehensive कि कोई difficult question ना आ जाए, but अभी तक तो कोई नहीं आया है. So no, you are absolutely right. The frequency of resistant mutations, I looked at it, is as good as rifampicin, as good as bedaquiline. And TB resistance is also very, very important to take into consideration. But since it's a combination therapy, it's less than the normal antibiotics like fluoroquinolones, Lelo, or any other antibiotic we use for that, because we are using 4 or 5 drugs in a cocktail to kill TB, and that the hope is that it doesn't lead to the resistance emergence.
So the second question I had was in terms of how much of activity you see with the flex pumps when you basically give this drug.
Yeah, we haven't done that experiment as yet. We haven't done. I think that's a very nice question. We haven't done that.
And efficacy of the drug, what are the side effects, if at all?
I think the side effects.
Bedaquiline.
You mean bedaquiline?
Does this drug has any side effects, or
which drug? The six.
So the bedaquiline. I mean, is there any way where you can modify, or is the work going on so that you can use lesser of the concentration and then?
Till now, bedaquiline has been used widely. And more than 1 million patients across the globe, across all the colors, ethnicities, races, and there have been no major discontinuations. Of course, there will be side effects, like as in some people, diarrhea ho jata hai, headache ho jata hai, nausea ho jata hai. There was a major concern few years ago of cardiac arrhythmias. But that now with the latest phase 3 data, with the latest data, global surveillance data, that is not really needed. So initial thing was ECG monitoring of the patients, but that is also not really done nowadays.
Thank you.
So, do you think in long-term combination therapy of the bedaquiline with J&J 6410 is required?
That's my hope, that we can remove the linezolid. So the hope is that we can remove the linezolid with 6640 or any other drug which behaves in that, and we create a regimen for patients which can be treated in 3 to 4 months and without any side effect.
But in long term, like purine, once you inhibit the purine biosynthesis, so there won't be precursor available for the ATP synthase to work. So bedaquiline, how it will work at that time?
We don't know that. We don't know how it will work in the long term. It will only once we have the patient data, we will come to know. But the aim is that because bedaquiline is synergizing with this purine metabolic inhibitor, that synergy drives the shortening of treatment duration.
Sir, my question may not be as scientifically sound as others, but
I was also not scientifically sound. Don't worry.
Sir, just out of curiosity, I want to ask that Amos law considers inflation, that since 1900 you showed that the $1 billion shift. So does it consider inflation or not?
Did what?
Sir, inflation, the value of money decreases over time, sir.
Yeah, I don't know. That's a very interesting point. What is the role of, just for the audience, what is the role of inflation? I was just trying to project the slide here, but what is the role of inflation in Erome's law? Any economists here? Or jisko mere se zyada economics ki knowledge ho? Anybody who can answer him?
Sir, the second question
Now,
is...
wait. Let me check the audience. Am I the only, or हम सब एक ही इसमें तैर रहे हैं? खड़े होके बताइए यार. You are the only one. You should come to the stage and give the answer. Are you happy with the answer? हाँ समझ आया क्या बोला मुझे तो कुछ नहीं समझ आया decrease in the purchasing power.
Actually, it's like suppose we can purchase some consumables at 1 billion in 1900, say 1000 quantities. In 2000, we will be purchasing only 100 of them or 10 of them. So basically, we are reduced with the purchasing power. So that's why our research is not as good over the time. So the second part I want to ask.
Offline. जब कभी आंसर नहीं आता है ना स्टेज पे the best way is to say offline. हम इसको offline लेते हैं.
So my second part is that you showed one data that 1 million bedaquiline was given and 5 million was prevented. So can you shed some light how was that data generated or presented?
So there are a lot of epidemiological studies. So there are a lot of studies. These are statistical models which people use that if you have 1 million infections prevented, means treated or in 1 million patients, how much that prevention will lead, how much that treatment will lead to prevention. So there are a lot of epidemiological modeling and data which goes into that. So definitely there is, you can find those things. These are very routinely done. Huh? Irum's law is adjusted for inflation. Thank you. Thank you. So, okay.
That is another question, Bhavtosh. So for your bedaquiline, is it only specific to MTB or it also work to the other intracellular pathogens?
Bedaquiline is only specific for MTB. Bedaquiline has not been used for any other indication. So most of the treatment of bedaquiline has been for MDR TB, or it is indicated for MDR TB. It's not even indicated for extrapulmonary TB. But doctors use their medical judgment in terms of seeing what's the best use of bedaquiline.
Thank you.
Yeah. 1 minute.
Expenditure.
If you are prescribing as a doctor or as a patient, I think Government of India or most of the national TB programs give bedaquiline for free. So it's freely available to all the patients across India as part of their national TB programs. And the government takes care of all the charges. That's why I said that we have to be very optimistic in terms of how we see TB, because of the role which government plays in terms of reducing the incidence, in terms of nutritional support, in terms of freely available medications that goes in TB treatment.
Anil, just continuation of that. Here. Look at the other side.
Sorry.
So just in continuation to that, because now you have developed 2 different drugs, you have so much of experience. So according to you, on an average, how much it takes in terms of money to develop a drug from the scratch?
You mean to say till what stage?
I mean, till the stage of a clinical trial, if I can say so.
Till a stage of clinical trial, there are regular.
And if I talk about this specifically for a TB, because
there are regular parameters, matrices which are very industry-wide applicable.
Okay.
So usually till phase 1, we say between $20 million. It's not very much big amount of money we talk about, 20 to 25 or maybe 30. That's the maximum you can reach till, that's on a very higher end. You can do it on a very low budget. But usually the challenge is that you need to bring everybody on to the single program very, very focused. So the teamwork is the key more than what you need in terms of the money. Money is secondary. I think there is money available to do TB research anywhere in the world, but to create that team, bringing the chemists, toxicologists, clinicians, all on the same platform and working every day for the next 5 years, that's the key.
Anil, one more question. So I was wondering, what data do you have for the new drug with respect to the persistence?
We don't have much data. But we have data in the publication on different heterogeneous TB populations, like in different starvation media and all that. We may have some data on dormancy, but I think it is out there, but we don't have full data on the uh, persistence, say. Yeah, I think, yeah.
This particular drug, what was the one is, of course, the value of money, but is there anything apart from value of money, since you had a lot of philanthropy and public funders also on the
Yeah, I think I'm sorry about I didn't talk about the funding. Most of the program, like Bedeclin, we had a lot of funders. Means we had alliances. I said that USAID was very much instrumental in terms of generating that supply chain. I think if THSTI discovers a new drug, you really have to think about how do we build the supply chain for those particular medications. For here, we had USAID, but for 6640, we had a TB Alliance, not as a funder, but as a partner. We had Gates Foundation, TBDA. They have this very nice consortium. They are now pushing these kind of molecules further in the pipeline. And yeah, I think most of the other work was through small fundings here and there, but those are several small funders which get involved during the course of all TB programs. It's not only the PUREP, because we did a lot of other things which will also be highlighted in the next few years, but I think, yeah.
And if you were to develop any other drug, would such partner composition, you would see any different than this?
I think partners come along the way. What you need is a champion. Everything for any particular successful program, Manish, very good at. You have been in industry, you know how the dynamics work. But for anything, whatever we do in our lives, we need one person who's accountable for running and pushing the program. So you need a champion. You need champion. I call it 6 C's. We need champions. We need people who work in the team. We need I won't go in the 6 C's concept. Somebody told me, what is the 7th? They said that you forgot the 7th C. I said, what? They said, cash. So yeah. So you need you need one person who is accountable for that, who believes in the story. Okay.
So, sir, is there any hope for using monoclonal antibodies against TB?
Yeah.
And are there any products in pipeline or?
I think we can always use monoclonal antibodies. People have used monoclonal antibodies for HIV. People have tried to use monoclonal antibodies for RSV. For viral infections, this has been commonly used. People are using interferon treatment for hepatitis B. So there are a lot of antibodies which have been used in the space of infectious diseases. Of course, in immunology, the whole immunology research is driven by the monoclonal antibodies. But in TB, the challenge is that we need to go away from injectables to orals. So we don't know at what stage an antibody can be developed as an oral molecule. I am sure that in next 10, 15 years, we will have formulations where you can give antibodies orally. There will be science. There is a lot of everybody in the world is looking for antibodies which can be delivered orally, or even peptides which can be delivered orally. So I feel that, yes, if there are antibodies which can be delivered orally, surely I think that will be very, very nice to have. But we are still, according to the national TB programs and WHO, we need an all oral regimen. Why oral? Because TB, as we know, goes to patients which are in socially, economically backward. So somebody who has to go to hospital every day for 6 months, they lose part of their earning livelihood. एक रिक्शा पुलर को बेचारे को हर दिन हॉस्पिटल जाना पड़ता है just to get an injection. Part of his day is gone, he's finished. So that's why the oral treatment, not only from the safety and the compliance perspective, but also from the economics, from a patient side perspective is very important. Okay, I think
most of the countries, they like a very few institutions they do on TB research, like outside developed countries you can see. But what I want to say, like how the public health, majorly can be improved so the TB could be eliminated. So most in like India, how?
yeah, the question is how will the public health be improved? Public health is a very complex thing. As Rajesh yesterday pointed out, it's not only the treatment. It's a composition of the environment. It's composition of what you eat. So nutrition is a very, very important aspect of public health. It's like the amazing increase in the cardiovascular disease in India is really, really, if you look at the way TB grew in 1960s, 70s, it's same thing happening with the obesity and cardiovascular disease. And that's also a public health issue. So that issue cannot be solved by medications alone. You have to do nutritional interventions at different stages, change the dietary, be more strict with food companies, see what you are, and raise the general awareness in terms of prevention of cardiovascular disease, because you can prescribe statin to everybody, but in the long term, from a public health perspective, preventing hospitalizations, it's very, very important that you take other measures rather than relying on therapeutics alone. And that's why government is very keen on improving the nutritional status of children with TB or patients with TB.
Thank you.
Hello? Sir, one last question. This side. So what is the oral bioavailability of both these drugs? And also why it cannot be given through IV due to some toxicity or something, or something else?
Bedaquiline is orally available. It's very nice, fantastic orally bioavailability, very long half-life. It stays in patients for very long, almost 3.5 months terminal half-life. So that's why it's very, very powerful. 6640 of some of those who are chemists here, the oral bioavailability is low. So we need to increase these kind of molecules, we need to increase the oral bioavailability. So that's something which we are working and a lot of other people are working. Thank you, everybody. Thanks for listening.
Thank you very much, sir, for that insightful and inspiring lecture. You've not only taken us through the scientific journey from bedaquiline to J&J 6640, but also given us a compelling vision of what the future holds for tuberculosis treatment. Your work continues to inspire the TB research community and reminds us of the impact that science can have on global health. We're truly grateful to have you with us today and for sharing your deep knowledge and experience. Thank you, sir. The Honourable Minister, Dr. Jitendra Singh will soon join us. He's currently visiting the MRC facility. Meanwhile, we're going to play a video showing our upcoming MRC Medical Research Center and Translational Research Facility.
At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions.
May I have your attention, please? The Honourable Minister, Dr. Jitendra Singh ji, will be joining us shortly. So I request you to kindly be seated. I'm pleased to announce that the Honourable Minister is just right outside the auditorium and he'll be joining us shortly. The Honourable Minister is just outside the auditorium, so please be seated.
not a very long journey in the terms of
We are glad to announce that
times.
the Honourable Minister, Dr. Jitendra Singh ji, is now amongst us to celebrate this special day with the THSTI family. I now request our Executive Director, Professor Ganesan Karthikeyan, Professor Rajesh Kokhle, Secretary at DBT, Dr. Alka Sharma, and Dr. Jayanta Bhattacharya to kindly escort the Honourable Minister on the stage for the lamp lighting ceremony.
वरदे वरदे वीणावादिनी वरदे 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 वीणावादिनी वरदे प्रिय स्वतंत्र रव अमृत मंत्र नव प्रिय स्वतंत्र रव अमृत मंत्र नव भारत में भर दे वर दे वर दे वीणा वादिनी वर दे काट अंधुर के बंधन स्तर बहुजननी ज्योतिर्मय निर्झर काट अंधुर के बंधन स्तर बहुजननी ज्योतिर्मय निर्झर कलुष भेद तम हर प्रकाश भर जगमग जग कर दे वर दे वर दे वीणा वादिनी वर दे नवगति नवलय ताल चंद नव नवल कंठ नव जलद मंद्र रव नवगति नवलय ताल चंद नव नवल कंठ नव जलद मंद्र नव 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 के नव विहग वृंद को नव पर नव स्वर दे वर दे वर दे वीणा वादिनी वर दे वर दे वर दे वीणा वादिनी वर दे
Ladies and gentlemen, as we gather here to celebrate 16 remarkable years of science, service, and societal impact at THSTI, it is only fitting that we begin by hearing from the person who leads this vibrant institution with clarity of vision, compassion, and an unwavering commitment to translational excellence. We're deeply honored to have with us today Professor Ganesan Karthikeyan, Executive Director, BRIC THSTI, a renowned cardiologist and Professor of Cardiology at the All India Institute of Medical Sciences, New Delhi. Sir, I now request you to deliver the welcome address, followed by the presentation of the annual report, which captures the Institute's achievements, milestones, and momentum over the past year.
No, nothing. No. Good afternoon, everyone. And on behalf of the THSTI family, I wish to welcome our Honourable Minister, Dr. Jitendra Singh. When people say normally that people have taken time out from their busy schedule, he actually has taken time out from his busy schedule. He needs to leave quickly. So I will just cut short my welcome. So all of you are welcome here, and we welcome our Minister as well, and Dr. Rajesh Gokhale and Dr. Alka Sharma. And we will skip some of the things. I know all of you have come all the way and stayed overnight for this occasion for signing the agreements. So we'll have that done in the presence of the Honourable Minister, and then he'll speak to us for a few minutes. Thanks.
Thank you, sir.
We hope to keep the momentum in the coming years as well.
We organized our second industry business meet, Singh, in NCR Biotech Cluster on the 14th of July, 2025. We are elated to present that the event has led to several collaborations across various domains. We now invite our industry partners in order of announcement to join us on the stage for the exchange of collaborative agreements. Beginning with the vaccines from Serum Institute of India Private Limited, we welcome on the stage Dr. Umesh Shaligram and Dr. Sachin Kale for collaborative agreement on development of controlled human infection model to test novel influenza vaccine candidates. From Indian Immunologicals Limited, we welcome to the stage Dr. Priyabrata Patnaik and Dr. Sridhar Kota for agreement on development of multivalent poultry vaccine. From Primas Biotech Private Limited, we welcome to the stage Dr. Prabodha Kundu and Dr. Nupur Mehrotra for agreement on evaluation of the safety and efficacy of influenza vaccine candidate. In the domain of cell therapy, from Immunil Therapeutics Private Limited, we welcome to the stage Dr. Lakshminath Gokinda and Dr. Gopinath Jakka for the agreement in the field of cell and gene therapies. From Artemis Education and Research Foundation, we welcome to the stage Dr. Sudeshna Kar and Dr. Rucha Chandavaskar for agreement to establish an indigenous end-to-end CAR T cell platform. From Miltony Biotech Private India Limited, we welcome to the stage Dr. Priya Hingorani and Dr. Sivakumar Natarajan for the agreement on cell manufacturing training program. From Agilent Technologies India Private Limited, we welcome to the stage Mr. Nandakumar Katahil and Mr. Sandeep Rastogi for agreement on training and application support services at THSTI facilities.
In the domain of monoclonal antibodies, from Origin Pharmaceutical Services Limited, we welcome to the stage Mr. Akhil Ravi and Ms. Ruxana Khatun for agreement for advancing innovative biopharma pipeline for monoclonal antibodies. From Genex Genomics Private Limited, we welcome to the stage Dr. Ashwin Kashikar and Ms. Supriya Kashikar for agreement for technology development for monoclonal antibodies. Balman Eswaran Biomedicine and Materials Science Private Limited, we welcome to the stage Dr. S.V. Eswaran and Dr. Sujit Kumar Thakur for an agreement for development in drugs and antibodies against tuberculosis. From Danaher India and its affiliate Cytiva, we welcome to the stage Mr. Sanjay Murdeshwar and Dr. Ashish Tejam for agreement for fostering bioinnovation and biomanufacturing in India. From Dodo Health Private Limited, we welcome to the stage Dr. Shantanu Pathak and Dr. Kashma Kothari for agreement for advancement of maternal and newborn healthcare outcomes through predictive modeling. In the domain of biotherapeutics, from Sandotia Nimandis Pro Bioceuticals Private Limited, we welcome to the stage Mr. Varun Sureja for agreement for research and development of probiotic and microbiome therapies. For the promotion of investments from Indian Private Equity and Venture Capital, we welcome to the stage Dr. Amit Pandey and Capacity Building and Ms. Yachna Luthra for agreement for promoting bio-based deep tech innovations in the cluster. For establishment of Center of Excellence, from Waters India Private Limited, we welcome to the stage Mr. T. Anil Kumar and Mr. Vikas Vora for agreement for establishment of Center of Excellence for Drug Metabolism and Pharmacokinetics. From Becton Dickinson India Private Limited, we welcome to the stage Dr. Subhadeep Mukherjee and Mr. Sumit Khurana for agreement for establishment of Center of Excellence for Flow Cytometry and Single Cell Analysis. And finally, from Dr. Dang's Lab LLP, we welcome to the stage Dr. Naveen and Dr. Leena Chatterjee for an agreement for repository services at THSTI.
Make it small, yeah, make it short. Quick, make it short. All right, all right.
THSTI परिवार के लिए यह अत्यंत हर्ष और गर्व का विषय है कि हमारे बीच भारत सरकार के विज्ञान और प्रौद्योगिकी मंत्रालय के राज्य मंत्री स्वतंत्र प्रभार, पृथ्वी विज्ञान मंत्रालय के राज्य मंत्री स्वतंत्र प्रभार, प्रधानमंत्री कार्यालय में राज्य मंत्री, कार्मिक लोक शिकायत और पेंशन मंत्रालय में राज्य मंत्री, परमाणु ऊर्जा विभाग में राज्य मंत्री और अंतरिक्ष विभाग में राज्य मंत्री माननीय डॉक्टर जितेंद्र सिंह जी की गरिमामय उपस्थिति है।
Now, Dr. Karthikeya was saying that पिछली बार आपने इनकी तारीफ कर दी थी, तो मैंने कहा कहीं उस तारीफ के चलते आज मेरी तारीफ में लंबा समय ना ले ले. So I am hard pressed for time. So, but I'm sure each one of us knows each other so well, so we hardly need to introduce ourselves, not write those matrimonial-like columns. Anyway, so having said that, it's a great pleasure being once again over here in one of these premier institutes. Dr. Rajesh Gokhale, our colleague and secretary in the Department of Biotechnology, Dr. Alka Sharma, Dr. Karthikeya, Dr. Pramod Garg, and members of the faculty, students. And when Dr. Karthikeya proposed that we're going to have this program on the 16th, I didn't realize that nobody could actually, at that point in time, foresee that it would clash with the timing of the mission that is happening over there with the International Space Station. So incidentally, the timing so got matched, and now like the docking with the International Space Station had about 40 minutes earlier. So also it's going to happen 30 minutes earlier. So I have to rush back. And that's precisely what the media persons asked me, what's it? I said, had it been a domestic airline, we could have boasted of a before time arrival. But now I'm worried about my arrival being there in time. So I can't even ask them to have a delayed arrival. So nevertheless, I think this is one of the institutes which has actually come of age in a very beautiful way, but is often not talked about much, rather underrated one, though it's being one of the only ones of its kind in the country. And it has actually created a number of success stories which have given India a global recognition. If we talk of the vaccines, the trials done over here, but I was telling Gokhale that you have to learn to beat your drum, because others who made money out of it even went away with the Padma Awards, whereas your team was hardly referred to. So hereafter, for each trial, after every phase, we'll try to summarize it, just as we've done in case of another trial that we did in collaboration with the Christian Medical College, the first phase of the genetic therapy for hemophilia. And not only did it get published in the New England Journal of Medicine, which is perhaps the oldest journal, the Lancet, the New England, and the BMJ. And at the same time, we also made a good showcasing about it. And it's not only for the sake of publicity, but also that those who are the stakeholders for whom you're doing all this work should get. And in fact, I've tried this is everywhere across the institutes in the country. We have about 37 institutes with the CSIR alone, which is also another wing of the. And there too, they were working in the institutes, and the people next door didn't know that they were working for them. For example, there was an institute under Dr. Kalai Selvi which was dedicated to aromatic plants, the herbal medicines, and they were doing beautiful job. They were also carrying out trials on resistant anti-tuberculosis drugs, etc., etc. Which sometimes you're at a loss to find the allopathy answers. And they had they are very successfully launched a Flori mission, also an aroma mission, come out with that so-called Purple Revolution, and a new genre of over 3,000 lavender startups, making lucrative profit out of it, but nobody knew. So I said, I'll come only when you have a farmer's meet, an agricultural's meet. And they managed to gather about 200 of them. And I said, today we will not speak, no lecture, nothing. Let them do the talking and tell us how they feel, being with us, seeing us for the first time. Do we look the same, or we have two eyes and three noses or whatever? And the same strange kind of observations came. One of them said, साहब हम तो यहाँ से गेट से सामने से रोज निकलते थे, हमें मालूम नहीं था अंदर क्या है. दूसरे ने कहा, साहब हमको किसी ने पूछा हमने कि यहाँ क्या. बोले, साहब ये यहाँ पे तो अंदर वैज्ञानिक होते हैं, मेढक काटते हैं. और एक तो ये भी कह बैठा कि शाम को 5 बजे के बाद यहाँ से नहीं गुजरना, क्योंकि दिन को उन्होंने काटा बाड़ी की होती है तो रात को भूत लाते. So, and I think the kind of experiments being done in this institute, that reputation is more easily possible to be emanated with the kind of trials being done over here. So that's why I said we have to, because if ultimately it is for the society, for the ease of living and the kind of therapeutic and prophylactic answers that you are discovering over here, need to reach out to the people. So it's not only for public. So I think we should, as I said, this is the medical center that I went around just now is perhaps one of the first of its kind in Asia. And we need to tell around the people about it. The other day I was in one of the agriculture universities, and a student along with a guide came to me. And she said that for her completion of a thesis, she wanted some more inputs, for which she needed to go to a certain country where she's applied, and she wanted help to facilitate that. And her guide also said so. And I was taken aback, and I told them all this work is happening in one of the institutes under Dr. Rajesh Gokhale, that Genomic Plant Institute. And neither the guide nor the student were aware of this. So if that is the kind of outreach, then I think there is something amiss on our part. But nevertheless, I think this is also one of the best times happening for biotechnology, because and you get the sense from the counseling sessions that happen after class 12 and also after NEET, and many of the boys and girls now opting for biotechnology as one of the preferred subjects, which was not so till a few years ago. If you randomly ask somebody what is biotechnology, he would not be able to figure out whether it's biology or technology, engineering or medicine. But now they got to understand. And at least in case of Shubhanshu, also been telling for the last 1 week, almost every day, that he's conducting his life-size experiments with the kits provided by the Department of Biotechnology. Even yesterday I said so. So I think we need to tell them so that they can make the best use of us. And it's all evidence-based, because all of us sitting in this room have been taught to speak with evidence, with statistics, not randomly, like sometimes the political jargon may be. It's not so. So if you go that way, we have figures. We were just about 10 billion bioeconomy before this government came. But after the Modi government came in, now we have more than 170, and we hope to be 300 in the next few years. Our startups were just 50. 50, sometimes embarrassing saying so. We are now 11,000 biotech startups. And the huge lot of industry representatives over here are a testimony to the kind of potential they find in this sector. And they would be knowing better than me, or be also understanding better than me, that the world over, among the economists, now the view is the next industrial revolution is going to be the bioeconomy driven, just as the last one in the 1990s was IT driven. And now we have also come out with a structured, institutionalized mechanism, like, for example, BIRAC, which most of the industry representatives who are referring over here, is one of the success stories, like in space. So we have an institutionalized structure to facilitate public-private participation, to determine the nature of collaboration, the scale of collaboration, the entities of the collaborators, both the private and the public sector. So I think we are proceeding even that in a very scientific manner. And we also need to tell people so that they make best use of it. The National Research Foundation, I think, is a better version of what we already had in US. In fact, the team from US headed by Punch was with us when we developed our own model with the Indian ethos also incorporated. And just last week, that 1 lakh crore fund for RDI is again dedicated to the industry because primarily. So we are lucky that we have a political dispensation which has no inhibition, no skepticism, no reservation in involving the private sector. And that's how we move forward, because if we need to have a global role for ourselves, or if we say to be a global player, we need to follow global strategies. We compare ourselves with NASA without realizing that NASA doesn't depend on the government alone, and so also. And the mutual skepticism has to be given up. And as far as this institute is concerned, you had the vaccine from here, of course, even the HPV, and I'm glad that the HPV team is also there, coming in a different collaboration now. Yeah, over there, Poonawala. And as was mentioned, the monoclonal bodies and the preclinical network laboratory, and of course the medical center, research center, which we just went around. And I must compliment the director for this meeting that you had last, I think 1 day back, yesterday you had, so which could bring them all together. And when I called him up just before reaching, I said, I'm running short of time, I have to go back. So he said, we'll skip the visit to medical center. I said, skip everything else, but not the medical center. That's for my own learning as well. And then he said, he also put across a condition. He said, then you don't have to skip meeting the industry leaders because they have stayed overnight. But I don't know whether they stayed overnight just to see my face or to sign the agreements. But nevertheless, whatever it is, collectively it is for a good reason. And therefore you're welcome. I don't have to, I can go on and on because all my life I've been more of a keynote speaker and a faculty rather than, performing the ceremonial task of lighting the traditional lab and quitting. But since the time constraint is beckoning me, I wish to take leave of you to be back again, because it's always a good educative experience being here. I wish I had more time to do so, actually. And I just leave today with the hope and wish that the Department of Biotechnology and the biotechnologists of this country will be the spearheaders of the next industrial revolution, the bioeconomy revolution of India, which would also be carrying ramifications for the rest of the world. So you have a great sterling role to perform in the next few years. You have an opportunity to be the torchbearers of the next economic world, not only domestically, even globally. Thank you very much.
धन्यवाद महोदय। मैं हमारे एग्जीक्यूटिव डायरेक्टर सर से अनुरोध करता हूं कि वह हमारे मुख्य अतिथि डॉक्टर जितेंद्र सिंह जी को स्मृति चिन्ह भेंट कर उनका सम्मान करें।
I now request industry guests, government officials, and THSTI faculty members to kindly move to the third floor terrace for lunch. I request the remaining guests to move to the auditorium terrace for lunch.
leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. A sustained pipeline. Clinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council under the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research, areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research, areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research, areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council under the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research, areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research, areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council under the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research, areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Look at this. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research, areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing
Check. Check. Hello. Check. Mic. Check.
5 नंबर प्ले करो 5 मानस लिखाओ
Hello. Hello.
थोड़ा और प्लीज साउंड
Hello. Hello.
यस
Hello. 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 La 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 जीवन के दिन छोटे से हम भी बड़े वाले कल की हमें फुर्सत कहाँ सोचे जो हम जीवन के दिन फुर्सत कहाँ सोचे जो हम वाले। तैयार है सर? हाँ, दरवाजा बंद कर दीजिए प्लीज। जी कर सम ये खूबसूरत जी सम ये खूबसूरत चार पलों से सत कहाँ सोच जो जी लुकी हम सत कहाँ सोचे जो जिंदगी ये ये जिंदगी ये जिंदगी जाने ये दिल जोड़ना जीना जिसे आता है वो दिल में मौज मना ले कल की हमें फुर्सत कहाँ सोचे जो हम ला 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 कल की हमें फुर्सत कहाँ सोचे जो हम मत सही है। थैंक यू। 4 o'clock कुछ और है क्या? नहीं सर है ना आपके लिए कैसा लगा सर? सुना नहीं आपको? इसमें इंटरनेट ऑन है आपकी? चलो ऐसा सुनो। हाँ सर, आप पूरा नहीं गा सकते।
चेक 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 हेलो चेक चेक 1 चेक चेक
Hello. Check. Hello. Hello. Check. 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 One. 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 Check. Hello. Check. 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 Hello. Check. One. One. Two.